The human body relies on two primary communication networks to maintain homeostasis. While the nervous system is hardwired using direct electrical connections, the endocrine system acts as a wireless network. It consists of anatomically disconnected glands secreting signaling molecules directly into the bloodstream to manipulate distant tissues. Mastering this network requires coordinating multiple biological disciplines at once. You have to track the systemic physiology of organs, the cellular feedback regulation, and the molecular structure of the signaling molecules themselves. The central rule for understanding these signaling molecules is chemical determinism. A hormone's chemical classification dictates its physiological behavior. Look at this diagram. Peptide hormones are water-soluble and cannot cross the cell membrane. They bind to extracellular receptors, triggering an internal signaling cascade. The receptor converts ATP into cyclic AMP, a second messenger that activates protein kinase A, causing massive signal amplification. Because they act through these transient second messenger cascades, the clinical rule for peptide hormones is their effects have a rapid onset but are short-lived. In contrast, steroid hormones are derived from cholesterol. Being lipid-soluble, they easily cross the cell membrane to bind with intracellular receptors. Upon binding, the complex enters the nucleus and binds directly to DNA, altering the transcription of specific genes. By directly altering the amount of mRNA and protein present in a cell, steroid hormones exert effects that have a slow onset but are long-lasting. There is a third category, amino acid derivative hormones, modified from single amino acids. These require overt memorization because their chemistry is less predictable. Catecholamines bind to G-protein-coupled receptors, like peptides, while thyroid hormones bind intracellularly, like steroids. A hormone's chemical structure dictates its mechanism of action, which in turn predicts how fast, how deeply, and for how long it will change the body's physiological state. The central command structure of the endocrine system begins with the hypothalamus. This organ acts as the primary bridge linking the nervous system to the endocrine system. This illustration maps the hypophyseal portal system. It is a direct blood vessel connection that allows the hypothalamus to secrete compounds directly to the anterior pituitary. Hormones released here travel down the pituitary stalk and bind to receptors in the anterior pituitary, stimulating the release of other hormones without entering the systemic circulation. The anterior pituitary synthesizes and secretes 7 different products, which can be categorized using the mnemonic flat peg. The FLH hormones, FSH, LH, ACTH, and TSH, are tropic hormones, meaning they cause the release of another hormone at the organ level. The PEG hormones, prolactin, endorphins, and growth hormone, are direct hormones, which act directly on non-endocrine target tissues. To maintain a strict homeostatic set point, this master axis uses a negative feedback loop. When a downstream target gland releases its hormone, that product travels back to inhibit the upstream releasing hormones in the hypothalamus and anterior pituitary. There is one critical exception to standard releasing factors. Prolactin inhibiting factor, which is actually dopamine, is released by the hypothalamus to decrease prolactin secretion. A drop in dopamine removes this block, allowing milk production to begin. Looking back at the anatomy of the pituitary, the posterior section operates entirely differently. Rather than a portal blood system, the posterior pituitary connects to the hypothalamus via the axons of nerves. A major point of clinical confusion is synthesis. The posterior pituitary does not synthesize any hormones. It contains nerve terminals that simply receive and store 2 hormones produced directly in the hypothalamus, antidiuretic hormone, or ADH, and oxytocin. Oxytocin also breaks the rule of negative feedback. It operates on a positive feedback loop. During childbirth, oxytocin promotes uterine contractions, which stimulates more oxytocin release, driving a mechanism that amplifies contractions until the definitive endpoint of delivery. The hypothalamus-pituitary axis functions as the body's master control board, utilizing precise physical pathways and feedback loops to maintain baseline stability across the entire system. The thyroid gland at the base of the neck contains 2 distinct cell populations with dual functions, setting basal metabolic rate and regulating calcium. This diagram illustrates the classic negative feedback loop of the thyroid axis. The thyroid's follicular cells produce triiodothyronine and thyroxine, known as T3 and T4. These hormones dictate metabolic activity and provide negative feedback to both the anterior pituitary and the hypothalamus to decrease TSH and TRH secretion. We can observe what happens when this loop breaks down by looking at this bar graph of TSH levels in mutant mice. The PV mutant mouse expresses a thyroid hormone receptor that cannot bind T3. Because the receptor cannot recognize the hormone, there's no negative feedback. The brain compensates by secreting enormous amounts of TSH, causing levels to skyrocket. Returning to the thyroid, the parafollicular cells, or C cells, handle the gland's second function, producing calcitonin. A helpful mnemonic is that calcitonin tones down plasma calcium levels by increasing excretion from the kidneys, decreasing absorption from the gut, and increasing storage in bone. This flowchart charts calcium and phosphorus homeostasis, detailing the push and pull between calcitonin and its direct antagonist, parathyroid hormone. PTH is produced by the parathyroid glands, which sit on the posterior surface of the thyroid. Following the green stimulation arrows, we see that PTH raises blood calcium by mobilizing calcium from bone and increasing renal reabsorption. It also activates vitamin D, which is required for the absorption of calcium in the gut. Critical physiological parameters like serum calcium require the precise competitive push and pull of antagonistic pairs to maintain a strict homeostatic set point. Perched on top of the kidneys are the adrenal glands. The gland is divided into the outer adrenal cortex and the inner adrenal medulla, a distinction that denotes completely different functional classes of hormones. The adrenal cortex secretes corticosteroids, which are steroid hormones divided into 3 functional classes, easily remembered as salt, sugar, and sex. Salt refers to mineralocorticoids like aldosterone. Aldosterone promotes sodium reabsorption in the vistal convoluted tubule and collecting duct of the nephron. Water follows the sodium cations, increasing water retention and raising blood volume. Sugar represents glucocorticoids, namely cortisol and cortisone. Cortisol mediates the body's slow, long-term stress response. It raises blood glucose through gluconeogenesis, providing a steady source of fuel during times of prolonged stress. Sex refers to cortical sex hormones, including androgens and estrogens. Certain enzyme deficiencies in the adrenal synthetic pathways can result in excess androgen production, causing severe developmental abnormalities like ambiguous genitalia in genotypic females. Nestled inside the cortex is the adrenal medulla. Unlike the cortex, the medulla is a derivative of the nervous system and produces amino acid derivative hormones known as catecholamines. These catecholamines, specifically epinephrine and norepinephrine, trigger the fast, short-term fight-or-flight sympathetic response. Epinephrine rapidly increases the breakdown of glycogen to glucose and shunts blood flow away from the gut, directing it to the skeletal muscles, heart, lungs, and brain. The adrenal gland physically encapsulates the body's entire stress apparatus, divided by anatomy into slow, long-term steroid responses in the outer cortex and fast, short-term amino acid responses in the inner medulla. The pancreas houses small clusters of hormone-producing cells known as the islets of Langerhans. These distinct cell populations act as the command center for blood glucose homeostasis. In the fed state, rising blood glucose levels trigger pancreatic beta cells to secrete insulin. As a peptide hormone, insulin binds to surface receptors on target tissues, stimulating the immediate uptake of glucose from the blood to facilitate anabolic processes like glycogen and fat synthesis. During a fasting state, alpha cells secrete glucagon. Glucagon is a catabolic hormone that raises blood glucose levels by triggering glycogenolysis and gluconeogenesis, causing the liver to break down stored fats and glycogen and dump glucose back into the bloodstream. A third population, the delta cells, produces somatostatin. This hormone acts as a master inhibitor, suppressing the secretion of both insulin and glucagon. When this system fails, dangerous hyperglycemia occurs. In type 1 diabetes, autoimmune destruction of the beta cells halts insulin production entirely. In type 2 diabetes, the body produces insulin, but end organ receptors become insensitive to it. Both conditions leave glucose trapped in the bloodstream. Beyond metabolism, there are a few remaining targets. The gonads are stimulated by LH and FSH from the anterior pituitary. In response, the testes produce testosterone, while the ovaries produce estrogen and progesterone. Finally, deep within the brain, the pineal gland secretes melatonin to regulate circadian rhythms. The pancreatic regulation of blood glucose acts as the ultimate antagonistic axis. By constantly toggling between anabolic storage and catabolic release, it ensures the brain and body always have the exact fuel they need to function. The endocrine system operates as a highly integrated logic gate circuit board across the entire human body. To decode this system, start by identifying the hormone's chemical classification. Knowing if a hormone is a peptide or a steroid tells you its receptor location and whether its physiological effects will be rapid and short-lived or slow and long-lasting. Next, track the negative feedback loops. If a downstream target hormone is abnormally low, the logic gates dictate that upstream releasing hormones from the hypothalamus and pituitary must be elevated to compensate. Finally, locate the antagonistic partner. Whether it's insulin pushing against glucagon to balance blood sugar or calcitonin fighting parathyroid hormone for calcium control, the full homeostatic picture always involves a push and a pull. The endocrine system's architecture is built on chemical rules and feedback logic. Mastering this biological machine provides a durable framework for